I am Dr. Monica from NIT Pharmacy Institute. I am taking the subject Pharmaceutical Microbiology, which code is BP303T. In this Pharmaceutical Microbiology, in my last lecture, lecture number 1, I have discussed about the history part. I discussed about the definition part, that is what is the definition of microbiology, what are the various branches which we deal with in microbiology, and I tried to cover the history part, in which history part I covered 2 generations, 2 eras. One was discovery era and second one was spontaneous generation theory. So, in history part, as I told, I covered discovery era in my first lecture and spontaneous generation theory in my first lecture. In that lecture, I told you 2 scientists worked. Now, 2 scientists were, one was Robert Hooke and second one was Antony van Leeuwenhoek, who discovered the microscopes, and once they discovered the microscopes, they were able to find it out what are the creatures, they were able to see those microorganisms. And in spontaneous generation theory, what scientists did, scientists actually worked on the condition that whether the microorganisms are coming from living creatures or these microorganisms are coming from non-living things, or whether they are coming from air, whether they are coming from soil, whether they are coming from that is what we call as food material, or there is some living creature within the environment from where the microorganisms are generating. And the scientist who worked under spontaneous generation was, that is first Francesco Redi, who was against this spontaneous generation. He was trying to tell you that microorganisms can't generate spontaneously, they generate from the living beings. Until and unless there is no living creature, microorganisms will not generate. After that, Johann Needham worked, after that various scientists worked, and some were in favor of spontaneous generation and some were against spontaneous generation. But the only scientist who proved that microorganisms come from living things, they can't come from the non-living thing, was Louis Pasteur. He worked under the golden age of microbiology, and by his experiment he tried to prove that until unless our non-living thing will not get contaminated with the microorganisms or the living creatures, until unless they will not be contaminated, no microorganism can grow. Where once they will contaminated, once the living creatures come in contact with those growths, yes, microorganisms will grow, and living beings are generating the that is microorganism. Biogenesis is the process by which microorganisms are being generated. After that comes the germ theory. In golden age, in this lecture I will try to focus on the golden age, I will try to focus on the germ theory, I will try to focus on the chemotherapeutic age, how the scientists generated antibiotics, how they find it out that germs are the cause of disease, how they find it out that we can kill germs with the chemotherapeutic agents. So, these eras we will try to focus in the lecture number 2, that is golden age, in which many scientists work and sort out many problems, find out the solutions to many problems. Germ theory disease, in which scientists try to find it out that germs are the reasons, are the causative agents which are causing diseases, and we have to find out some solution to kill those microorganisms, those species. And in chemotherapeutic age, the scientists were able to find it out, the chemical agents by which we can kill the microorganisms. So, first scientist who worked under golden era was Johann Tyndall. Now, who was Johann Tyndall? Johann Tyndall was the scientist who find it out of one of the sterilization process by which we can kill the microorganism. But as we all know, we can kill microorganisms with the help of heating. In the previous years, doctors, scientists were using heating as a process to kill the microorganism. We just try to boil our water to kill the microorganism. We always try to boil our milk to kill the microorganism. So, heat was the main process by which microorganisms were killed in the previous era. But he was the he was the scientist who discovered heat resistant bacteria. Heat resistant bacteria are the bacteria which can't be killed by that is heat. Heat process can't kill those bacteria, those species, and that structure he named as, later named as endospores. Endospores are the agents by which, are the species, are the parts of bacteria which can't be killed by heat. Heat se kill nahi kar sakte hum unhe. So he was the person, once he find it out those heat resistant bacteria, heat resistant microorganisms, heat resistant creatures, that is endospores. So he demonstrated the process, he discovered a process by which we can kill. And how we can kill? He find it out a sterilization process by which microorganisms can kill. How he demonstrated? He used the alternate process of heating and cooling. That means heating is being done, that means at alternate processes heating is provided. We will heat the product, after that we will cool, we will heat, and after that we will cool. In this way 5 cycles will be repeated, 5 cycles in which repeated 5 cycles can kill all the endospores, even all the heat resistant microorganisms even. So alternatively we have to kill, we have to heat, we have to cool the product, so that even the endospores which are present within the sample gets killed, and we will obtain a sterile media, we will be able to get a sterile product. So alternate process of heating and cooling was given by Johann Tyndall. He was the scientist who gave that process of Tyndallization, and he worked under the age of golden era. After that, Louis Pasteur. He was he is actually the father of what we call as pharmaceutical microbiology. He is the person who gave lot, and he did many experiments, he found out various techniques and worked on various type of microorganisms. He was the person who gave the term fermentation. He was the person who told that fermentation is the process of converting carbohydrates to alcohol or organic acids using microorganisms. Before that, what was happening? Before that, scientists were saying that fermentation is a process which takes place due to chemicals. Chemicals are the reasons, chemical changes are taking place to generate this process of fermentation. Fermentation is not generating due to any living creature. They were saying this is a chemical process, enzymatically or biochemically they are converted to the, that is fermented, the food gets fermented. But he was the scientist who proved that the fermentation is a process which occurs due to microorganisms such as yeast or bacteria under anaerobic conditions actually are responsible for this process of fermentation. Without microorganisms, without yeast, without bacteria, we can't ferment any of the carbohydrate, we can't ferment any of the that is food product. So for fermentation microorganisms he named as ferments. Ferments are, he was the person who concluded that fermentation is catalyzed by vital force called ferments within yeast cells. There is a vital force within the microorganism, there is a vital force within the yeast cells which is responsible for fermentation. The ferments were thought to function only within living organisms. Alcoholic fermentation is an act correlated with life and organization of yeast cells, not with chemical process. So fermentation is not a chemical process, it occurs due to microorganism, it occurs due to yeast, it occurs due to bacteria. If yeast is present within the sample, within the food products, only then fermentation will take place, only then the food will get fermented. Carbohydrate will change to alcohol, carbohydrate will change to organic acids in presence of microorganisms. After that, he also gave the process of pasteurization. Now, what was the process of pasteurization? Pasteurization is a process in which foods are treated with mild heat. Means, as we all know, sterilization is a process in which heat is provided and we kill the microorganism by the process of heat. But as we all know, when we heat any of the product, any of the food product, if we heat, then the nutritional value will be lost. All nutrition, as when we heat any of the product, nutritional value will be lost and we will not get any nutrients if we heat the products to kill the microorganism. So, that is why Pasteur gave a mechanism in which we require only low heat, that is less than 100 degrees centigrades, to eliminate pathogens, to kill the microorganisms, to extend the shelf life of the product. So, pasteurization or Pasteur gave this process of pasteurization in which mainly food products are heated less than 100 degrees centigrade to eliminate pathogens, to kill microorganisms, to kill pathogens which can cause disease. So, pasteurization of milk widely now practiced in various countries. In several countries we are practicing this pasteurization technique, in which generally the products are heated to 60 degrees centigrade. Now, we do not require to heat them to 100 degrees centigrade, we only need to heat them to 60 degrees centigrade, but one parameter was extended, that parameter was time. That means you have to heat at 60 degrees centigrade for 30 minutes. If we see at home, we always boil our milk for just when the milk get boiled, we just stop the gas. So, 100 degrees centigrade there was time was not considered, but in case of pasteurization he mainly considered two parameters, one was temperature and second one was time. That is, temperature he kept less than 100 degrees centigrade and time he extended for even 30 minutes. As you can see, two parameters, 60 degrees centigrade maintained for 30 minutes, or alternatively heating to a higher 72 degrees centigrade, 72 degrees centigrade for 15 seconds. We will obtain a sterilized form of product. Sterilized product we will obtain if we increase the time span and we reduce the heat. Heat is reduced and time span has been increased, and yet higher temperatures are available for shorter periods to get a pasteurized product. Today pasteurization is used widely. It is used widely in various industries like dairy industries. We are using the process in various food processing industries. So, pasteurization was one of the technique which was designed by Louis Pasteur. That credit was given to him, that is Louis Pasteur. After that come the germ theory of disease. The germ theory of disease also was credited to Pasteur in 1864. What he established when he was discovering the fermentation process, he was able to find a technique, he was able to find it out that disease are also caused by microbes. He developed a relationship between microbes and disease. He was the person who introduced the germ theory of disease, but he was not able to prove by his experiment how germs are causing diseases and how they are responsible. The scientist who actually worked that in the germ era or in this era of germ theory of disease was Koch. Robert Koch. In 1843 to 1910, he was the main person who related germs with disease, that germs are the main causative agents which can cause disease. So, how he proved that microorganisms or germs are responsible for diseases? He performed an experiment, or even that experiment is being used in our, that is, pathological labs today also. Today also that practical which he designed in the era of in the era, that is in 19th century or from 1843 to 1910, he was the scientist who proved, he developed an experiment, and that experiment is being used nowadays also in the pathological labs. Now, how he did? He tried, and first point he considered was the causative agent must be present in all affected patients. First he worked on the affected patients who are suffering from the disease. So, he tried to find it out what is the reason behind that disease, or if any of the person is ill, any of the human being or any of the animal, any of the person is ill, that patient is being affected by any of the microbes. So, he first worked on the affected patients. He tried to take out their blood samples, and in the blood samples he tried to find it out the reasons behind the cause of that disease. So, when that the agent must be capable, and once he tested the blood of patients, he found it out some agents, some causative species were there which was causing the, which was were not found in the normal patients, or normal human beings are not having those creatures, and those creatures he named as causative agents which are present in the patients, but which are absent in the normal human beings. So, the agent must be capable of being isolated, and he tried to find it out that we must be able to isolate that particular causative agent from the sample of blood or from that patient. He found it out, the techniques of isolation, the techniques by which we can isolate those species from the culture or from the blood samples or from that particular patient, and that agent must be capable of being isolated and cultured in pure form. Means I have already told that pure form means single creature, means single species, single causative agent must be there. We must be able to isolate that single creature from the blood samples, and we must be able to develop their pure cultures हम उनके develop कर सकें. And when the isolated cultured agent is introduced into healthy beings, and once those has been at first he found it out the agent which is present in the patient, second he tried to isolate that particular causative agent. After isolating that particular causative agent, what he did, he introduced that culture, he introduced that isolated pure species in the healthy beings, and this, if that individual creature is creating the same disease in the patient, in the healthy human being also, same disease is being regenerated within the healthy beings, then it is said to be a causative agent, then it is said to be an agent which can cause a disease and which is responsible for the disease in that patient. Patients were having that disease because of that isolated pure culture when he introduced into the healthy beings, and the same causative agent must be isolated again from the host. Means once as we all know, we have to repeat our process not once, we have every time we need to repeat our process at least 3 times to validate the reason, to validate our process, until and unless we have not repeated and that results are not regenerating with same results are we are not able to obtain, we have to move on, we have to validate our processes. So, that process is being validated also, was repeated 3 to 4 times, and the same isolated pure species was injected, was that is introduced into the healthy beings, and at least if all the healthy beings in which that particular causative agent has been introduced is generating the disease, and the healthy beings are getting those same disease reoccurs in the all individuals. So, that we say that this is a particular causative agent which is responsible for a disease. As we all know, in today's era also COVID is a virus is, until and unless we are not knowing that that virus is present in the patients, and from the patients we are obtaining and that samples, and from samples we are trying to isolate that that is virus, and after that we try to those viruses are being injected within the normal beings, within the animals, and scientists try to find it out that these viruses, what impact they are causing to the healthy beings, what impact they are causing to the healthy animals. So, once this is the mechanism how the scientist found it out whether the causative agent is there, this is the only agent which is causing the disease. Means first you need to isolate, second you need to inject into the healthy being, after that you need to find it out that same disease is being reoccurring within the healthy beings also due to that particular virus, due to that particular bacteria, due to that particular organisms. So, causative agents are being evaluated by these postulates, these are what we call as Robert Koch postulates. 4 postulates are there. 1, causative agent need to be found in the affected patient. Second, it must be able to be isolated, we must be able to isolate that that is particular causative agent. Third, we will introduce that particular causative agent into the healthy individuals, and same disease must reoccur. If the same disease is reoccurring, then only we will state that this is the particular microorganism which is causing the disease. So, after that, after Koch gave the Koch postulates, the Joseph Lister. Now, once it was found that in the previous slides we saw that how germ theory got generated, how scientists tried to prove that germs are causing disease, germs are responsible for causing disease. Koch developed the Koch postulates. So, other scientists were working along with them. The one scientist's name was Joseph Lister. Now, who was Joseph Lister? Joseph Lister was a scientist who worked in the era, that was 1827 to 1912. What he did? He is actually a doctor. He was a doctor. He was working, he was doing surgery on the patients. So, what he did? He found out a chemical agent. He is acknowledged with the title known as father of antiseptic surgery. Father of antiseptic surgery means he was the person who generated chemicals, agents which can be used before surgery, which will not allow the microbes to enter into the that is patient when the surgery is being going on. So, antiseptics were developed. Antiseptics were first discovered by this scientist known as Joseph Lister. He is acknowledged as the father of antiseptic surgery. Joseph Lister's contribution paved a way to safer medical procedures, means as surgeries were going on, many patients were dying because of when the surgery goes on, they get contaminated with the that is what we call as microbes. Microbes enter the that is body, or because of which further diseases start reoccurring. So, that is why antiseptic play a vital role before the surgery. He was because of the discovery of these antiseptics, he was able to dramatically decrease the deaths of child. He was able to do surgery in which the in which the patients will not be affected by the microbes. Microbes will not be able to enter the human beings and will not be able to enter the blood during the surgery, because of which lots of deaths were going on at that moment. He was also able to generate a process which is known as sanitization. We are recently also we are using sanitization everywhere. We are trying to sanitize, we are trying to sanitize our hands, we are trying to sanitize our floors. So, sanitization, chemical agents was first developed by this scientist named Joseph Lister. So, that is why he is also known as father of antiseptic surgery. After that comes Edward Jenner. Now, who was Edward Jenner? We have studied about the diseases, we have studied that many scientists worked on the chemical agents, and he was the first person who studied the immunology, that our own system can fight against a disease, our own body can fight against a disease. We have such agents, we have such antibodies within ourselves, that those antibodies when generate will cure the infections. We will be able to generate antibodies, we have the immunity power. So, he was also Edward Jenner was a scientist who is known as the father of immunology. That means, he was the person first who found that immunity, that immune system, that our immune system can work and can fight against the disease. What observation he found? He found that the persons who are working, who are milkmaids, who are working with the cows, and who are dealing with the cows, and they are milkmaids, and they are the persons who are if get infected by the smallpox, they will not generate a disease. He was the first person to observe that thing, that the person who works with the cow will never be affected by a virus known as smallpox. So, what hypothesis he generated from the observation? From his observation, he was able to find that if any of the person is intentionally being infected by the cowpox, will become immune to smallpox. If the person hypothesis was that the person who already had a cowpox disease, who already have survived because of the cowpox disease, will get impacted by the smallpox, because antibodies generate within that person and antibodies will cure that person. Experiment what he performed was, he inoculated an 8-year-old boy with cowpox matter from a blister on the hand of an English milkmaid. After 4 months, he attempted to challenge the cowpox inoculation by exposing the same boy to smallpox virus. First, he worked with a boy, he injected that boy with the cowpox lessons, and so that the antibodies generate within the child, and once the antibodies will generated, after that, after 4 months, he tried to inoculate the smallpox virus within the same child, and he proved that antibodies got generated and smallpox will never affect this child, this child will never fall ill because of smallpox, because antibodies have already been generated in that child. So, result demonstrated about smallpox immunization, that the immunity got created not because of smallpox virus, but because of cowpox virus. The persons who were dealing with the cows got immunity towards the disease known as smallpox. Immune system got developed, and those persons are never being affected by the virus known as that is smallpox. So, immunity really works a lot. That is why we always focus on our immune system, we try to improve our immune systems. So, in this era also we are trying to that is increase our immunity power, so that we can fight with the diseases. So, immunization or immunity plays a vital role in fighting with the infections. So, immune systems must be strong of the patients, or that immune systems can be increased by even inoculating with the same that is, if we say cowpox was the virus which got inoculated in the person and antibodies got generated. So, immune system we need to work on it, and we need to improve our immune systems. After that came the that is scientist named Paul Ehrlich. Paul Ehrlich was a scientist who got a Nobel Prize for medicines. He was the agent, he is known as the that is father of chemotherapy. He was able to find it out the chemical agents which can act as a magic bullet, which can cure the patients with the help of chemicals. Salvarsan was the medicine he discovered. He was the person who discovered methylene blue dye can act as a magic bullet. It will selectively kill the cells which are causing diseases. It will selectively kill, that is why it is also known as magic bullet, because only infectious pathogenic microorganisms got killed with those that is chemical agents. So, that is why he got a Nobel Prize for developing the chemotherapeutic agent. He worked in the emerging field of serum therapy. As we all know, plasma therapy is going on, serum therapies are going on. So, he was the first person who developed this that is serum therapy, antitoxins and chemotherapeutic agents. After that, Alexander Fleming, he was the first person in 1928 who generated a wonder drug known as that is penicillin. He was the person in 1928. Fleming was working with a other species known as Staphylococcus aureus. He was working on Staphylococcus aureus, but accidentally what happened, that Petri plate containing Staphylococcus aureus got contaminated with a new microorganism known as Penicillium notatum. He was working on Staphylococcus aureus, but when he came after vacations back to home, he found that his Petri plate which was having Staphylococcus aureus got contaminated with the microorganisms known as Penicillium notatum, and that Penicillium notatum was generating a what we call as a chemical agent that was responsible for killing those Staphylococcus aureus species. Penicillium notatum was generating a agent named penicillin which was killing that is Staphylococcus aureus. That is what we call as antibiotics, which are generated from some of the species, from some of the microorganisms, and are responsible to kill the other microorganisms. That what we call as antibiotic, and he was the first person to generate penicillin which can kill Staphylococcus aureus species. So, after that, in the last I will just like to tell you about the 20th century, what scientists are doing right now. They are finding new antibiotics, they are finding new antifungal agents, they are finding new vaccines, they are exploring the reasons to spread antibiotics. Even nowadays we all know resistance is being developed against antimicrobial agents. So, scientists are working a lot against this also. They are finding the reasons behind the antimicrobial resistance, why our body, our human beings are becoming immune or are becoming resistant to the antimicrobial agent. They are not going, the antimicrobial agents are not impacting or are not effective enough to kill the species or kill the microorganism or to cure the to cure the disease. So, resistance is being developed, though this in this era most of the scientists are working in this era, that is they are exploring the reasons for the spread of antimicrobial resistance. So, these are the various agents, new agents which are being developed in the era 20th century. These are the antibiotics. After that, many vaccination innovations have been done. We have find it out dengue fever vaccines, we have find it out the Ebola vaccines, AIDS vaccines, and nowadays we are finding the vaccine against the COVID also. Scientists are working on to find it out the vaccine for the COVID also. So, resistance on scientists working on the resistant parts also, why resistance of human pathogens to antimicrobial agents is being developed or vaccines is developing. So, scientists are working on this also. So, these are the references which you need to follow, and after that in this I am over with the history part, that is history of microbiology from the starting of 13th century till 20th we have covered, that is the history of microbiology, and these references you need to follow for this. Thank you so much. Thanks a lot.